experimental science, we tend to want to test and apply basic theory to real world problems, and we're really interested in establishing cause and effect. So it's a different approach from going out and sampling and monitoring for patterns and trying to infer cause. We're trying to establish it experimentally, and the artificial reefs have given me a great opportunity to do experiments on a scale that you wouldn't normally find in university research. So figure out which direction this goes. All right, yeah, I want to acknowledge some folks that have really been helpful over the years, in sequence. The funding for all of this work, all of the reef construction has been funded by FWC, and they helped to do a bit of the research in the early 90s, but then Sea Grant got us started in the 1980s, and for the past 2 decades, National Marine Fisheries Service has funded all of the research activity. And I might add that with the NOAA Fisheries, none of that work was motivated by questions specific to artificial reefs. The artificial reefs were simply the study systems to study the biology and the ecology of the grouper. And so we're trying to weave these two areas together. And to give you an idea, the kind of level of activity I've had over the past few decades. In the 1980s, there was a pilot study done up here on Dog Island Shoals, and some work with stone crabs down in just off of Cedar Key. Got involved with some regional reef development in shallow water. Then in the 1990s, and established the Suwannee Regional Reef System, which is 22 reef arrays bracketing the mouth of the Suwannee River in about 40 feet of water. And then began in the 2000 planning and developing the Steinhatchee Fisheries Management Area with the first line of standardized reefs for monitoring the output of the Big Bend region, and then the 100 square mile triangle of the Steinhatchee Fisheries Management Area. That was enhanced in 2011 and 2012. So that's, we've been scaling up over this time period from experiments that were on the scale of 1 square meter plots and 5 square meter plots to now 100 square miles, scaling up as the questions have scaled up. All right, and the bottom line message from all of this work is summarized in that cartoon. What's it all about? Eat, survive, reproduce. Eat, survive, reproduce. That really is everything you need to know about what are we doing with artificial reefs, because every one of these animals, particularly the mobile animals, are looking to solve the equation of how do I find enough to eat and grow, and how do I avoid being eaten myself, because dead fish don't eat, they also don't reproduce. And then the bottom line is, if you're not reproducing, you're not really fulfilling your potential. So that's what it's all about. The difference is that every species is solving this problem differently according to their adaptations. And so the target species that we identify really matters, because the particulars of what we're accomplishing when we modify artificial habitat with artificial reefs is that we're trying to modify how they eat, survive, and reproduce. And so one size does not fit all, because every species has solved this problem differently. All right. The earlier work, the pilot study I mentioned, started off with a small crab that's only, at its biggest, is about that big across the carapace, and lives in cavities it creates in bryozoan colonies in seagrass beds. And so it was a good system for doing experiments where we could establish the animals are capable of evaluating the quality of the habitat, that is the size of the bryozoan and whether it would protect them from predation. And lo and behold, they were able to use those bryozoan colonies naturally occurring on seagrass beds as a means of not only avoiding predation, but securing mates. So the pattern of dispersion of the those refuges affected their mating frequency and mating opportunities. So that was a pilot study in advance of doing the same kind of work with stone crabs right here off of Cedar Key. And we scaled up from 1 meter and 5 meter plots to 300 meter plots using the same standardized modules for stone crabs as cavity modifications, and just altering the landscape. Oop, pardon me. See if I can get this to go back there. So, you know, the same number of modules in each of these experimental plots. And one of the things that we found was that, yes, the pattern of dispersion of the refuge affected the mating opportunities for the stone crabs. That was one of the things we were interested in. But more interestingly, the more widely spaced modules actually held a larger number of stone crabs and held them for a longer period of time than the more closely spaced modules. And that same kind of pattern of abundance with the same amount of refuge applied to black sea bass, gray triggerfish, and when we had an octopus invasion, you know, it applied there. So the same response by species from 3 different phyla were showing up. That's kind of a general principle or a general pattern. And we started going, well, why would that be? Well, all of these species take shelter in the structure, but then feed out away from the structure. And so we sampled the prey items that surrounded those reef materials. And there was a halo, as expected, around each of those reefs, commensurate with the kind of prey that these critters would eat. And we framed it up as a resource mosaic hypothesis. You got to make things sound really highfalutin to get them published, all right? And so the resource mosaic hypothesis, where the animals were responding to the landscape of resources where there was shelter as a central node, and then foraging out around that shelter. Carl Walters made the same concept much more famous by referring to it as a foraging arena theory, and it's become well established in population dynamics as accounting for how fish balance the food resources and the shelter resources, and the risk of predation and the need to go out and eat and grow. And so this really helped to establish one of the functions of artificial reef development is to make foraging grounds more readily accessible by altering the availability of shelter that's essential for these critters to avoid being eaten themselves. And so we then chose to scale that up and go from crabs, which were easy to handle and measure and so forth, to grouper, which are a bit more interesting for most of the anglers and the people interested in artificial reefs. And gag is a particularly good species for this for a number of reasons. One, it has what we call a spatially stage-structured life history. That means it's spawning offshore in the wintertime on the shelf edge break, and the larvae are transported inshore to the seagrass beds, and then they spend about 3 or 4 years as juveniles on the inner shallow shelf. Well, one of the nice things for us experimentally is that it meant we could do experiments in here with juvenile female gag, and we wouldn't have to worry about the messiness of reproductive behavior getting in the way of what are the trade-offs between feeding and sheltering, and how does the pattern of shelter availability affect the behavior. And so the Swanee Regional Reef System was built as a fully controlled and replicated experiment after 2 aborted attempts to set up experiments using culvert as material of opportunity and standard reef construction practices to set up experimental plots. And we found out we couldn't achieve the experimental controls we needed. And this may have been one of the first large-scale FWC funded projects using prefabricated modules as the reef material. Now that video is on one 16 cube patch reef with just over 150 gag, over half of which were above the legal limit. When these reefs were first put in place, they were not published. We didn't advertise their existence. To the best of our knowledge, they weren't fished. All right. And one of the advantages of the gag, as obvious here, is that they're a shoaling species. That's they're gregarious. They can achieve high densities, which is something that we were very much interested in. what's the density dependent effect on using shelter and finding food, compared to a red grouper, which is aggressive and territorial, and they're over dispersed. They spread themselves out on the same reefs. We would find numbers of gag like this, but only one red grouper per patch reef. All right. And so in the sampling, our original hypotheses that were developed with the stone crabs were borne out with black sea bass, white grunt, tomtate, other kinds of fish. But when gag came in numbers like this, we really focused on them as the target species, for all of the subsequent work. And what we were doing was we were manipulating, standardizing the reef unit for the quality of the patch, and manipulating the patch reef size and spacing. Now, from that, we were able to demonstrate that the gag exhibit density-dependent habitat selection. That means they choose habitat not only on the quality of the habitat itself, but on the densities of competitors for the resources that habitat provides. And of course, the resource value of the reef structure, through our experiments, was established as primarily shelter. The gag would occur in high densities, even at the expense of growth, if they had access to the shelter. Now, one of the other things, by virtue of having 22 reefs, each of which was a hexagon of a particular patch reef size and spacing, was that we had replicates that we could do an actual experiment on the effects of fishing. And so after a period of colonization, when we got the basic hypotheses tested, we went ahead and opened up some of these sites, published the coordinates, and made them available for public fishing. Within 8 months of that initial fishing effort, I think it was in November, and we did our sampling. 8 months later, the size for distribution and the abundance of gag on the published reefs were truncated, cropped right at the legal limit. I should say the reefs off of Dixie County, it was truncated just below the legal limit. All right. All right. Enough said. Yeah, but then over time, the reefs that were not published and made available to the public started to become discovered. And the rate of discovery depended on the patch reef size and spacing. Duh. Yeah. Smaller reefs are harder to find and harder to fish. And so that led us to thinking that, well, there may be a bottleneck in the life history of these of gag as they move across this shallow shelf. Let me go to this next slide here. One of the things we found was that the growth the growth of the juvenile gag on the smaller reefs was greater than it was on the larger reefs, and the growth on the smaller reefs that were widely dispersed was better than on the smaller reefs that were greatly clustered. Why? Because of the density of competitors for the food that was available in proximity to the reef, right? We also found that the gag, we put tags in their belly, surgically implanted, and monitored the residents on the reef as a measure of site fidelity, how long they stay on a reef. Did they stay long enough that we could attribute the growth to the particular reef? And as a way of looking at, well, what do they prefer? What's the best kind of reef? Well, experimentally, we can ask the fish to tell us. And so the gag actually preferred the larger reefs. Those were the reefs that, where they had the longest residence because of the greater shelter. And the reefs that were more isolated, by the same token, there would be fewer fish going out and competing for the prey surrounding the reef itself if those reefs are more widely dispersed. But they were having the better growth rates as juveniles on the smaller reefs. And by the fishing experiment, they were having the lower fishing mortality on the smaller reefs that were widely distributed. So that led us to think that, well, if we built small, widely spaced, unpublished reefs, that were harder to find and harder to fish, and which the juveniles had better growth rates, even if it wasn't the best reef from their perspective for natural mortality, we may be able to achieve higher growth and therefore better egg production out of the females once they join the spawning stock. So that kind of thinking gave rise to to the Steinhatchee Fisheries Management Area. But space use is something that's important. Let me just backtrack to this. During that telemetry study, we didn't do that as a mark recapture project. Those tags were internally implanted in the body cavity. There were no external marks, but out of the 81 gag that were surgically implanted with tags, we had 23 of those tags returned from fisheries catch. Yeah, what do you say, Mike? That's a tremendous fishing pressure. Yeah, when you have that kind of return rate. All right, but the tag returns, so those open circles right up there are where the Suwannee Regional Reef was, where this tagging study went on. Some of those fish were recaptured on the Suwannee reefs. We had one fish captured out by the spawning aggregation offshore. Those squares are the two marine protected areas, Madison Swanson and Steamboat Lumps, that are off limits because of the spawning aggregations. And a couple fish that were in between the Suwannee reefs and the spawning aggregations, but almost all of the recaptures occurred northwest of where those tags were originally released, suggesting that there may be a bit of a counterclockwise movement of the gag as they make that transition from juvenile habitat to the adult habitat offshore. But 9 months after it was last seen, we had one fish caught on an oil rig off Freeport, Texas. And 5 years after it was last seen, we had another fish returned from a depth and time of year where that fish was no doubt part of a spawning aggregation off of Veracruz, Mexico. And you don't normally think of a reef fish making that kind of movement. So, there's a lot of connectivity possible across the Gulf. The other thing here about their behavior, we normally think of putting reefs in barren sand bottom away from anything else. And that way we're not disturbing anything. But we were wondering, well, what happens if the landscape is more complex than a sand bottom? So we had the same kind of standardized reefs that were in a sand bottom represented by the yellow, and then in a fragmented live bottom habitat. This is off of Steinhatchee, same reef modules being there. And we had a telemetry array. This is dissertation work by Zy Biesinger, where there were receivers on the reef and 100 meters away from the reef, and the tags were implanted, so there was a signal being sent every 2 seconds by the gag during the study period, and we can monitor the locations by triangulating where the fish were. And one of the things that Zy found out was that in a fragmented live bottom, the gag foraging area was 7.5 times larger than it was when that same reef was located in a sand bottom. That's a substantial difference in space use, given the complexity of the landscape. And there's even evidence of fish setting up almost pathways where they move along the edge of the live bottom to move from one location within their home range to another area. And so, location. We have a hypothesis that over the long term, fish in this kind of environment may achieve a better growth rate than fish that are concentrating all of their activity right in the immediate vicinity of the reef. All right. So the Steinhatchee Fisheries Management Area, targeting 100 square miles, it was developed with 500 standardized reefs that were of the size that gave us the better growth rate. These are randomly distributed with at least 250 meters between patch reefs within that large area. We consider these conservation reefs. They're not published. It's peppered the landscape in there. And the idea was, if we could create habitat on a large enough scale in which the juveniles had better growth rates and better survivability, then that may improve the contribution to the fish stock. This was not a reef system that was built for the benefit of the fishermen. It was built for the benefit of the fish. Now, the northern portion of the management area Victor talked about that earlier, is designated for fishing reefs. And those are larger patch reefs that are better for fishermen because it puts more fish concentrated under the hull, because there's a larger reef on the bottom, more gag under the hull. But overall, you're not getting as many fish out of those kinds of reefs as you would if the same amount of reef material was more broadly distributed, right? So we monitored the reefs of the Swanee system, these evaluation reefs offshore, which are standardized reefs, the 4-cube reefs, the same as what was occurring in here over a period of time. So again, we have 15 years of data here, 10 years of data from these reefs. And from all of that monitoring, one of the things that we've come to the conclusion on is that the trends in abundance on the reefs is exactly what you would expect to see given the theory underlying how we think fish behave. You would expect that high quality habitat would always be occupied, all right? And lower quality habitat would fluctuate given the overall population abundance, right? So you remember the red tide of 2005 further south from here that shut down the gag fishery for a couple years because of all the mass mortality that resulted from that? Well, that gave us a natural experiment. We could see what happens to the abundance of gag on high quality habitat, that is habitat they prefer, and lower quality habitat, the less preferred reef types, over time. And sure enough, when the stock was declining because of that massive red tide in 2005, we saw the high quality habitat maintain the same abundance. The catch per unit effort on those high quality habitats with our diver counts stayed constant, despite having the overall population substantially reduced. The lower quality habitat lost abundance. In other words, fish abandoned lower quality habitat and were only found on the higher quality habitat. We saw the same effect with the red tide from 2014 after the population had recovered. The higher quality habitat also saw an impact in 2014, but it was nowhere near as great as the impact that occurred on the lower quality habitat. In other words, the fish would concentrate on the higher quality habitat and abandon the lower quality habitat when the population was low. And then they would spill over and reoccupy the lower quality habitat when the population was rebuilding. The other thing that happened is that we were monitoring the reefs here and the reefs out here, and they're of the same quality in both of those geographic locations, and the numbers were exactly the same. So for a given quality of habitat, you're going to get a certain number of gag in residence, all right? And so once they reached equilibrium between the inshore and the offshore reefs at the same habitat, we peppered this 100 square mile area with those conservation reefs and monitored what happened offshore. And as would be expected, these reefs inshore intercepted the juvenile gag moving out of the nursery grounds, and they occupied there. And we saw the reefs in this vicinity drop in their abundance. So when you have a large-scale reef development, as we did, you do end up with a redistribution geographically of the fish that are in the overall population. All right. Now, we were also sampling over the past few years inside the Steinhatchee Fisheries Management Area and outside to compare growth rates and survival of the fish inside and outside that conservation area. That's one of the big reasons why we built that management area in the first place. And what we found was that inside and outside, this was unfortunately the period immediately after the 2014 red tide, we didn't find an obvious difference in the growth and survival of gag inside and outside that management area. We go, well, wait a minute, that doesn't bode well for being able to use the reefs to enhance the survival and growth of the juveniles. But what we did find was that the inside we had an abundance of age 1 gag, and outside we had almost no age 1 gag. So one of two things or both things have to be happening. Either these reefs we're intercepting and concentrating the age 1 gag. Those are the fish that have just left the nursery grounds and moved into those juvenile shallow water habitats. And yeah, that's what we were wanting to do, was enhance, you know, those juvenile fish, right? And it could be that we have better recruitment of age 1 fish here than we do on either side of the management area. The other possibility is that we have equal recruitment inside and outside the management area, but the predation on those juvenile fish is greater outside than inside. Either way, that seems to indicate that we're able to have some effect of the biological benefits for the youngest of the juvenile fish. But that difference disappeared by the time they're 2, 3, and 4 years old, because we didn't see any difference in total survival or total mortality or total growth inside or outside. But one of the interesting things was the fish that were inside the management area had better liver condition and intestine condition, meaning their liver and their body cavity had more fat content than the fish outside, which tells us that they're in better condition. They're laying on better fat reserves for movement and overwintering and ultimately getting to a size where they could reproduce. But it's just not, during the time of our study, manifesting a faster growth rate that shows up in length of age. All right. So there is a possibility that there's a bottleneck in there. Now, what are some of the take-home messages for planting artificial reefs? Yeah. Bear in mind, eat, survive, reproduce. Whatever you're doing, you're modifying how the fish go about doing that. And from our work, paying attention to the shelter benefits and the foraging area surrounding the reefs is important. You know, the ecology can be pretty well understood and can be applied to reef design. All right. Identify the target species. What do we know about the behavior and the ecology? Because what you do for one target species may not be the best strategy for another. And the seascape or the landscape that you're modifying is as important as the reef type. Because when you put all your materials in one location, you're creating higher competition for the food that's available. So you may have an abundance of fish to catch, but the fish may not be getting the best benefit. It kind of sets up a tension here. You can design reefs for fishermen, or you can design reefs for fish, but you probably can't maximize both of those benefits simultaneously with the same reef design. Expect the hyperstability and catch per unit effort. The old attraction production question actually has merit. I think we've explained what the attraction is all about, all right? But it creates a situation where you can maintain a high catch rate in the fishery, even though the population is declining, and that's going to contribute to shorter seasons and smaller bag limits. All right, and one of the things we've come to realize is we know a lot more now about the fish than we do about the fisher behavior, and so Ed Camp, one of our newest faculty, is now trying to look at socio-ecological modeling of artificial reef systems, so we can get at what are the trade-offs between what the fish do and what the fishers do, and try to use that for helping to plan the best strategy for a given set of management objectives. And with that, if there's time, I'll take questions.
Yeah, we can probably take one question. So we can take one question. Yeah.
So under Restore Act, a lot of us have artificial reefs part of one of our projects. One of the things we have to do is decide what we're going to categorize artificial reefs under. If we categorize it under restoring fish habitats or improving fisheries, then I have to provide best available science to show that it does improve fisheries. Mm-hmm. And there's this constant debate there whether it's good for fish or whether it's not good for the fish. So which way do I go with that?
Well, when you refer to a fishery, it's the fish and the fishermen or fishers, right? So the work in the Panhandle, that was motivated, I believe, because of the economic loss and providing economic infrastructure for those coastal communities. So improving the fishery can mean greater fisher access, greater angler satisfaction, greater economic